I guess it's going to be the very first time that I'm going to be introducing the person who have taught me almost everything about research, which is actually Marta Grover. Marta is an associate professor in the department. She got a bachelor's degree in mechanical engineering back in Urbana-Champaign. She then went to do a master's in mechanical engineering at Caltech. Then she said, well, I will stay here for a little while, so she decided to do a Ph.D. in control and systems dynamics at Caltech. And as soon as she got the degree, she actually joined the faculty in the chemistry department in Georgia Tech. Her research covers many different multidisciplinary approaches. One of them is going to be here. And a few of her accolades, a few of her awards. She's an NSF Career Award recipient, and she in 2011 she won the Young Investigator Award of the Computing and Systems Technology Division of the American Institute of Chemical Engineering. And I would like to present the title of her talk, Prediction and Design in Chemical Evolution.
Morning, everybody. So today I'll be talking about a very collaborative, interdisciplinary aspect of my research. And so I just wanted to start off by highlighting the people who have participated in this research. So there are 3 postdocs who are part of this research, Sarah Mari Walker, Irina Mamadanov, and Andreas. Also a number of undergraduate students, so actually Jingya, Patrick, Daniel, and Garrett are all undergraduate students in chemical engineering. And the collaborators are Nick Hudd and Joe Shork. So Nick Hudd is in chemistry here, many of you know. And Joe Shork is a colleague of mine in chemical and biomolecular engineering. Aaron Engelhardt was a former Ph.D. student of Nick's. But all of this research was really catalyzed by the NSF NASA Center for Chemical Evolution. Nick is the PI on that center and has really brought this team together to work in an interdisciplinary fashion. So to start off, I just wanted to point out some basic underpinnings of research in origins of life chemistry and chemical evolution. So to just give a brief timeline, this is thanks to Nick Hudd. So the formation of the Earth occurred approximately 4.5 billion years ago. And the first evidence of single-cell life is around 3.5 billion years ago. Multicellular life appeared, or is first observed to have appeared around 600 million years ago, so much, much more recently. And then finally, the greater complexity of life today. So the research that I'm going to talk to you about is really aimed at understanding principles of chemical evolution that are needed to understand some of the early evolution prior to 3.5 billion years ago in the origins of single-cell life, and probably sometime after about 4 billion years ago, because the conditions on Earth were really too harsh during that first 500 million years. So research in origins of life and evolution can be divided up broadly into 2 camps. The top-down camp, so observing modern life today, looking at DNA sequences, bioinformatics approaches, as well as a variety of other approaches, but conducted primarily by geologists, geneticists, and archaeologists. Whereas the bottom-up approach tries to understand the principles of how life could emerge, how the transition from chemistry to biology could have occurred in the early Earth. And geologists are a very important part of that research to really try to understand what are the conditions on the early Earth, as well as physicists who sort of historically have done much of the mathematical modeling and theoretical work in this area, and organic chemists who are synthesizing molecules and observing how they react and assemble. There really haven't been too many chemical engineers in this field, but I'm a chemical engineer, so I put that on the list, as well as my collaborator, Joe Shork. But really the premise behind the bottom-up approach is that all forms of modern life, whether bacteria or humans, use DNA and proteins according to the central dogma of molecular biology. So information is stored in DNA, it's transcribed through RNA, and then translated via the ribosome into proteins, and then those proteins help provide a feedback loop modulating the replication of DNA. So the idea is if we can understand the origin of these functional biopolymers, that can help us understand more broadly origins of life. And the principles underlying chemical evolution that can allow us to then engineer responsive, intelligent materials that can learn to create better therapeutics, to treat diseases such as prion diseases or Alzheimer's disease associated with also principles of chemical evolution. But so specifically, I want to focus the talk today on the scope of the origin of functional biopolymers. Now, the Miller-Urey experiments, some of the seminal ones conducted in 1953, are ones that you may have learned about in your general chemistry class. And they established conditions for the abiotic, non-biological synthesis of both nucleobases as well as amino acids. So this is a photograph of that apparatus, and that's actually part of our center symbol as well. But a key open question is then how to make the functional biopolymers from these monomers, how to make RNA, DNA, and proteins, or molecules similar to them that then may have been refined through evolution over the years. Some of the challenges in synthesizing these biopolymers are the fact that without proteins, there are no enzymes. So modern synthesis, biochemical synthesis relies on enzymes. Another problem is called the water problem, but all of whether the linkages of nucleobases or proteins, they're made through condensation polymerization. So a water molecule is released every time a bond is formed. If you're conducting these reactions in the ocean, in water, then that really drives the backward hydrolysis reaction. So that's a big challenge. And then another challenge is the dilution issue. If you think about the volume of the ocean and how many nucleobases may have been present in the early Earth, that the system is very, very dilute, and again, that makes it very difficult to drive forward the reaction. So there are some possible solutions that have been proposed, many solutions, and these are some of them. None of them have yet really provided a definitive solution to this problem, but all of them could potentially play a role. And I think that's one motivation for the use of mathematical modeling or complex systems kind of analysis is that there are many competing factors, and really understanding the quantitative trade-offs is difficult to do with an intuitive or a reductionist sort of approach. But some of the possible solutions are considering mineral catalysts, so reactions on mineral surfaces as opposed to enzyme-catalyzed reactions. One potential solution to the water problem is to consider cycling conditions, so looking at perhaps tidal environment, tidal pools. So during part of the day, the system would be wet, but during other parts of the day, it would be dry, and that could allow the reaction to go forward during the dry time, and especially if we think about dew forming at night and then evaporation during the hot day, the colder conditions associated with the wet phase could then reduce the hydrolysis that would occur when it's wet, and then the hotter conditions during the dry phase could really drive forward the polymerization. Another possible solution to the water problem is to consider alternative solvents. Perhaps it was a urea-based solvent, not water, in which some of these reactions occurred in the early Earth. To address the dilution issue, surfaces are one potential solution, and they even could be mineral surfaces. So surfaces can concentrate molecules through attractive forces, but then they also could potentially serve as catalysts. Dispersed phases are another possible solution to the dilution problem. So instead of just assuming that all of these oily monomers, perhaps oily monomers, are equally dispersed in the ocean, that we consider something more like an emulsion, in which we have oily phases as well as wet phases, and that can really concentrate the system. And then the final point here says alternative chemistry. So all of these other points could be considered in the context of modern nucleobases and amino acids and biopolymers. But perhaps early in evolution, there were modified forms of information storing molecules and functional catalyzing molecules. And then so there are variants, perhaps, that would not have all of these same sorts of difficulties. Well, first I wanted to talk to you about a study. That was a collaborative study with postdoc Sarah Amari Walker and Nick Hudd. So Sarah has recently accepted an assistant professor position at Arizona State. And the idea here is to really understand the conditions, the chemical parameters, the environmental conditions that would allow us to build a system that can generate and sustain diverse molecules. So this is really, there are a couple of theoretical challenges or open issues in chemical evolution. One is that many of the mathematical models for replicating biopolymers start off with a diverse pool of sequences. But over time, that pool is really dwindled until one sequence comes to take over the whole system. And that's the sequence that can replicate the fastest. So it makes sense, and it corresponds to sort of the survival of the fittest narrative, single winner. But it's really not consistent with the importance of generating diversity so that open-ended evolution can continue. Also, if we want to build a system that can have complex function, we probably need more than one biopolymer to create a cascade of reactions. So that's one key question that this study was aimed to address, was how to have coexistence of diverse sequences. And then the other question was how a functional biopolymer that emerges in this soup, primordial soup, if it has a function, how it could benefit from that function so that it could gain a competitive advantage and make more copies of itself to really take hold in the system. Without cellular compartmentalization, it's very difficult to sort of keep the fruits of your labor. So you make some sort of catalytic product that's beneficial, but then it floats away. And so everybody benefits from it equally. So then that particular functional sequence that sort of randomly, spontaneously emerged in the system isn't really able to continue to copy itself. It might go extinct. But what we really wanted to do here is to understand the relationship between microscopic parameters, rate constants, and diffusivities, and then the system level properties. And that's kind of a theme throughout the different projects that I'll be talking to you about today in my role as an engineer and as someone who does system level complex systems modeling, is that we want to elucidate these relationships that are not intuitive from the outset using mathematical modeling. So here is the model of the system. We're considering the cycling conditions between a dehydrated phase and a hydrated phase. So we can think of those as day and night, dew at night, dry during the day. And during the day phase, we have a formation of biopolymers. So we have monomers of type A and B. So instead of, say, 4 nucleobases or 20 amino acids, we'll just consider 2 here. And we allow the spontaneous formation of new sequences with rate constant Ks, and also replication. So here we have a particular sequence. It can make an exact copy of itself through a replication event with rate constant Kr. These are both second order rate constants in monomer concentration, first order in the polymer concentration. So it can make an exact copy of itself. And the key point here is that these reactions are dependent on the local resources. So if there's a lot of monomer around, then you'd have a higher replication rate. So all consistent with max action chemical kinetics. One thing you might be asking is, what about mutation? And so here we decided to consider a scenario in which the replication is really under more of a thermodynamic control instead of a more kinetic-based control that would have mutations. So that's not consistent with modern biology necessarily. but we're getting our diversity through spontaneous nucleation of these new sequences. But we could potentially also add mutation to this model, but it's not something that we considered in this study in the interests of having a minimal model with a parameter space that we could explore. Now, during the night phase, we don't allow polymerization to occur. There's water around, and we assume that's negligible, but we have hydrolysis, breakdown of polymers. And we also have diffusivity. So if it's wet, you can imagine that the system could reconfigure itself. And these simulations are conducted using a stochastic simulation method. The reason is that we may just have 1 or 2 copies of a particular sequence, at least on a particular site. And so we really can't make the assumptions of continuum assumptions that would be needed to use an ODE sort of formulation. Sure.
You may have stated this, but does A and B have to bond, or can A and B and B form?
They can form any arbitrary sequence. Okay. And so there's a lot that I didn't say about this. We kept it relatively minimal in terms of kind of how we define the model. We originally did intend to have specify different sequences for each molecule. We even wrote the code so we could put in any arbitrary sequence of AB, AAAB. But what we found is, for one thing, there were so many possible sequences, and our spontaneous nucleation rate is relatively low. Otherwise, we're just making random things all the time, using up all the resources in making new sequences and not making copies. And so it was highly, highly unlikely that we would ever make the same sequence twice through spontaneous nucleation. Right? So as a result, we decided to just label each unique sequence with a number, an ID. And so we actually don't track that. Now, in reality, that the replication parameters would depend on the sequence, and I'll talk later about a study in which we do consider different kinetics for each sequence. But here, we're just assuming that every sequence replicates with the same rates. We call this universal sequence replication. So the idea really is to say, even if all of these sequences have the same kinetics, how can we see heterogeneities in the system? And that's really going to come out due to the spatial distribution in the system. The limited diffusion is going to determine the fitness landscape, actually, instead of these parameters. But of course, these would all depend on sequence. But we're not going to get here into predicting sequence dependence and folding and function and replication, and that would be just sort of an impossible task to do that for many sequences. It's probably not accurate, right? So for that reason, in this study, we assume universal sequence replication. So from a kinetic and diffusion point of view, all the sequences are equally fit, but they're going to have differences in their copy number due to stochastic effects and spatial variation. Oh, here are some of the key features of the model, some of which I just alluded to from Valeria's question. We have these regular environmental cycles, reversible polymerization, so that's the hydrolysis, in which the monomers are released and can be then reused by other sequences. So the hydrolysis doesn't lead to just dead products. These can sort of eat each other. Okay. But that leads to really better utilization of the limited resources, limited amount of monomers that would probably be present in the system. We have a closed mass system with a finite monomer concentration. So these simulations are two-dimensional, so we assume everything's going on a surface with limited diffusion. We have these get our diversity, new sequences in the system through this spontaneous polymer assembly, and that allows us to explore sequence space to look for sequences that would have a function. We have this universal sequence replication, so all sequences have the same parameters. And then in the end, we look at the emergence of this functional polymer. So through most of the study, none of these sequences have a function. But then at the end, there's a function that emerges. So what you can see here is one simulation with a particular combination of the 5 parameters in the system. And early on, there's a transient in which the total polymer population grows from 0 up to this higher level. And then it looks like pretty much nothing's really going on in the system. But in fact, there's a lot going on in the system if you look at this left plot. That here are a few different sequences out of the many different sequences that were nucleated over 2,000. And you can see early on, when there's a lot of free monomer, we nucleate a lot of new sequences. Some of them really grow. Some of them go extinct. And then later on, we can also nucleate new sequences, but it doesn't happen as often because the monomer concentration is lower, because we have monomer in polymer. And then some of these sequences really do take hold in the population, these late emerging sequences. And then at the bottom, we can see spatial maps associated with different times. Early on, we have these very small clusters. Polymer concentration is at the top, monomer concentration is at the bottom. And then over time, we get more polymers. We can see these clusters are growing. And over time, they continue really to grow. So even though it looks like, again, here there's not much going on, we have a lot of dynamics in terms of our species and as well as this clustering phenomenon. And the clustering phenomenon was actually something that we didn't really necessarily expect to see from the outset, that it was something that we observed in the simulations we think could be important, but was kind of unexpected. So just to give you a little bit more intuition about that, I'm just going to show a movie. And this actually is not polymer concentration. This is a sequence similarity determines the color. So early on, we see these pockets form, these red pockets that have the same sequence. So just one particular sequence that's copying itself. But then you can see, if you watch maybe this area here, these clusters start to kind of merge, but they still maintain their separate concentration of sequences. So you might think of these sort of like organelles in a cell. But you can see there's also regions of non-red, so sort of overlapping sequences. So the sequence similarity is lower there. And that means that there's the possibility for cross-catalysis, some sort of interaction between multiple species that could lead to more complex function. So this is, in the absence of cellular compartmentalization before lipid bilayers, surfaces could provide kind of a rudimentary compartmentalization to create cell-like organization. So I already said that the goal here was to relate these sort of more microscopic parameters, chemical and physical parameters, to system-level metrics. So we defined 6 system-level metrics. The average lifetime of a species. So we want the sequences to be able to really take hold and potentially demonstrate their function and benefit from their function before they would go extinct. We also look at the average species population size, so how many copies of each sequence in the system, as well as the number of different species. So really we want all of these to be high, but of course they can't all be high at the same time. So there are quantitative trade-offs here that we're trying to evaluate. We also look at the total population size and the sequence exploration rate, as well as the average local diversity. So we'd like to have multiple sequences on the same site, at least in some places, to enable more complex function to ultimately emerge. Now, we were looking kind of for a sweet spot here in terms of getting system-level metrics in all of these areas that we could work with or live with. And this red dot is the one that we really like the best. So it has a replication rate constant of, I don't know, kind of in the middle of this range. We just vary by orders of magnitude. And then the hydrolysis rate constant is relatively large, 0.1. That means every night we lose about 10% of our polymers every night. And then the other 3 rate constants, the diffusion rate constants and the spontaneous nucleation rate, are held fixed in this particular set of plots. But you can see the trade-off pretty well in total population size versus sequence exploration rate. So we want to keep generating new sequences so that we can find new function, but at the same time we want to be able to carry over sequences overnight. And so that's why with this red dot you can see there's kind of a nice trade-off here. But we could have picked some of these other points that still maintain a similar ratio of replication rate constant to hydrolysis rate constant. But we go forward with this red dot here. We like that point. So then we move on to look at the role of diffusion. And these are images of the spatial patterning. So if this is the monomer hopping rate in the simulations, or you can think of it as a diffusivity, and the polymer hopping rate. So we really get very little polymer forming if the monomers and the polymers don't diffuse. We can't really utilize the resources because they're not able to move around. So if we raise the monomer hopping rate, then we get a lot more polymers and we have very small clusters. If we have a low but non-zero polymer hopping rate, we start to see the formation of these clusters. So if the polymers can't move at all, then everything just stays on its same site. The monomer can come in and out, but we really can't get clusters forming. Now, as the polymer formation hopping rate gets even larger, then we see the clusters, really they're becoming very diffuse here, but really on a larger length scale with a larger simulation domain, you would see larger clusters forming. But so really that's what the polymer hopping rate really determines kind of the size of the clusters. So this is really all that we can resolve in our particular simulation domain. So if again we look at the same 6 system level metrics, but now as a function of the monomer and polymer diffusivities, we see some different trade-offs. Actually, as long as the monomer hopping rate is sort of high, high enough, there's not a lot of variation in most of these system level properties as long as we have some polymer diffusivity. So the blue curve here corresponds to no hopping. But as long as we have a little bit that we can utilize our resources well to maximize the population, we get a relatively insensitive sequence exploration rate. So that's kind of the lesson here, is that we want the monomer concentration to be pretty high so that we can utilize our resources, and then the polymer hopping rate really doesn't matter so much in terms of most of these metrics. It does determine, though, the average species population size. So we might choose to have a higher rate there. That also gives us a higher average local diversity. It's just going to mean we have larger clusters in the system. So that, again, could enable more complex organization, organelles to occur. But we probably don't want to have a fully well-mixed system either. That's going to become important later for the emergence of our functional polymer, in which limited diffusion really helps a functional sequence take advantage of its particular catalytic function. And that's what's demonstrated here. So the hypothesis that was advocated by Ma and co-workers is that a first functional sequence to emerge in the early Earth may have been that of the monomer synthetase, a functional biopolymer that could create more monomer. So that's a much simpler function than, or that could activate a small molecule to become a functional a monomer. That's a much simpler function than, say, a polymerase that would march along and actually copy these particular sequences. So we decided to explore that particular hypothesis in that study. And what we found is that if we sort of arbitrarily give an emerging sequence this particular function of the monomer synthetase, of a late emerging sequence, that particular function, it can not only take hold in a population in which, if it had no function, you would have this blue curve for its population size. If we give it a function, it can really take hold in this population, even as a late appearing sequence. And it also has a much longer lifetime than the non-functional sequence, or even average all sequences in the system, including the early appearing sequences. So this azyme, we call it, the A enzyme, can really benefit from its function in this limited diffusion case. So this is a case of limited polymer diffusion. That was one of the lower levels that we considered. So here's then some spatial maps associated with that. So I think I like to call this set of images the It's a Wonderful Life. So it's this is what would happen if the functional sequence never appeared, like Jimmy Stewart in the classic movie. And this is what happens if that particular late appearing sequence does have that function, the azyme function. And then we also consider a later appearance of a B-zyme. So now we have a different sequence that appears at a different time in a different place, but it can make more B. And what we see here is that compared to, here's polymer, A monomer, and B monomer concentrations. We have clusters here, so we're making polymer. But if we have the azyme appear here, now we have this supercluster that forms with this region here that was bare, now has a lot of polymer there. But we also see that everybody benefited. So the idea here is that it's a combination of cooperation and competition, that you want to have both. So you want the azyme to be able to benefit the most from its function, but you'd like the system to benefit too. That means we can further create more sequences, further explore sequence space, increase that sequence exploration rate because we have more resources. Another thing you can see here is that we have a lot of excess A monomer and depleted in B monomer because we had more A than we could really use. But if we have this later appearing B-zyme, that now the entire population is much larger, and you can see that here more quantitatively with the total population with no functional sequences, the A-zyme, and the later appearing B-zyme really allows the whole population to really bootstrap up much more, because now we have a more balanced monomer concentration. We can really utilize both resources. So again, here's the movie form of that. So these are the same set of plots. It's a higher diffusion situation in this case, but we see that red spot now associated with the appearance of the A-zyme. So we have a lot more A monomer there. And then the polymers can also, those grow in those regions more as well. So we get more growth here as well compared to this polymer concentration plot over here. And here's the appearance of the B-zyme now. And so now that cluster can grow much more than this cluster over here. So the, I guess, conclusion from this study is that for an appropriately designed system, we can sustain diversity under this universal sequence replication assumption, with no particular function other than replication. And then if a functional sequence does emerge in that pool, because of the limited diffusion, it can benefit more from that function. It can take hold and propagate in the system, but we can also improve, the entire system can benefit as well as all sequences individually. So there are combinations of these rate constants that would allow us to see this kind of behavior, but I think the idea really is that if we're going to build this particular system, there are way too many things to try. And so if we can use modeling to help guide us into the right region of the parameter space, then we have more hope of identifying this sort of system level function. But we also understand that all sequences are not going to replicate equally. And so in this particular study, so this is with Andreas, we looked at moving beyond that universal sequence replication function and looking at different kinetic rate laws. And some of the key feature in this model, one is that we have this variable reaction order. So unlike the second order reaction that we assume for all sequences in the previous study, here we're looking at variable reactions order. So maybe some are first order events, others are second order events, others have observed reaction orders of 2.3. So we look at a range of different reaction orders that build in lots of different complex folding functions and diffusional limitations and all sorts of the complexities that we understand would really be occurring and would be sequence dependent. And in this case, we also consider an open system. So the previous system was closed to mass, but if we really want to have continued open-ended evolution, we really would like the system to be able to grow more like a biological system. And so we view this here sort of from the chemie perspective as a well-mixed reactor. So we're not going to consider diffusional limitations here. We're thinking more about what's happening on a particular local site in the system in which there is significant mixing. But we have replicators, different replicating biopolymers, as well as the J different food sources in the system, or monomers. And this model is formulated generally, but in this study we really looked at two monomer types, A and B again, and two replicators. So we're just looking at the competition between two replicators on the same site. And how can we, understanding the conditions under which we can get coexistence of both replicators, not have one dominate and take over the other. But we still, other important features are this recycling, this reversibility that allows the system to dynamically reconfigure. And so in this equation, we have a balanced equations for the concentration of each replicator and the concentration of each monomer in the system. So we have flow of monomers coming in and out. We assume that the replicators are more like cells. They're kind of stuck to the wall. They stay in there, and it can continue to grow. So you might think of this as a small pore in a rock in which the small molecules can move in and out. But this variable reaction order here we have on the monomer concentration allows us to have replication rates that are really context dependent. So depending on the monomer concentration, replicator 1 might be more fit from a replication point of view, or replicator 2 might be. So it's really context dependent in terms of which replicator is more fit. It's not just fixed. So it's more chemically realistic, but also brings in some additional dynamics into the coexistence question. So this is just an example of what were a typical simulation for one parameter set. So here's the replicator concentration of species 1 and species 2, and the monomer concentration. And one thing that we saw, one thing that we noted, so we considered 10,000 different parameter combinations. So we used a statistical method called Latin hypercube to really fill out the parameter spaces optimally with these 10,000 different parameter sets that include kinetic constants like rate constant prefactors, reaction orders, the composition of each A versus B in each particular sequence, as well as the environmental conditions, the inlet flow rates, and the inlet concentrations of the monomers. And what we found is that over these 10,000 different parameter sets, only 1% actually led to coexistence of both species. And this is important because it means that if you try two arbitrary sequences, they're probably not going to coexist. But yet this is still a vast parameter space. Over our 10,000 different parameter sets, we saw 100 different cases where there was coexistence. And if you're thinking about sort of searching through primordial soup, there would be combinations that could coexist of different replicators. So that was one of our findings. The other finding through the simulation study was that we see this linear growth rate profile in the replicators in the coexistence system. And then we weren't expecting this, and this is not what's usually seen in this sort of replicator modeling, but it makes sense. We're feeding the system. And so here the system is really then using up it as we're feeding it, and that's what's really limiting the growth. So as long as these are efficiently utilizing these resources, that it's not, the flow rate isn't so high that the monomer is just flowing out before it can be used, really this is something that we would expect and that is seen in, say, bioreactor fermentation systems during at least part of the growth phase. We also saw that we had a steady state in the monomer concentration. And these plots on the right are just zooming, it's a log plot so we can see the early transient. So early on, there's more going on than this very simple behavior, but then after that initial transient is passed and growth gets going, this is what we see. But based on this observation of the linear growth profile, we built a mathematical condition for coexistence in which we said, okay, let's assume each species has this linear growth and we have a steady state monomer concentration. What combinations of the environmental, composition, and kinetic parameters would allow us to have coexistence, meaning positive growth rate for both species? And so these plots map out that phase space in terms of the composition of the replicator. So if replicator 1 has 10 A units and replicator 2 has 5, then we have this green dot here, which corresponds to coexistence in the simulation. Now, if we then compare that to checking of these growth rate constants in our algebraic condition, we see that positive values for both, which again corresponds to the green dots here, are seen in both halves, actually, both of these quadrants. And so what we found overall over our 10,000 simulations is that our algebraic condition screens down from all the parameter space down to 2%. It identifies all of the coexistence parameter sets, but it also considers sort of a second shadow half of that parameter space symmetric, in which that linear growth solution exists but doesn't appear to be stable. But we still think that this is a really, really useful screen because it takes us down from all parameter sets to 2%, and half of those do exhibit coexistence. And we kind of understand why the other half we don't observe coexistence. So in this case, unlike the previous study, we get decoupling and therefore diversity, sustained diversity, not through limited diffusion, but through compositional balance. So what we've observed is that if both replicators have the same composition, we always have a single winner scenario. And that's why we have sort of these quadrants sort of over here. But this is something that we had observed with a balanced inlet condition. So we feed it equal amounts of A and B. But what we found with our algebraic condition is that any kinetic and compositional combinations of the two replicators can demonstrate coexistence with an appropriate inlet condition. So if we feed it different ratios of monomers. So it's not really about having certain molecules that can coexist or not, but it's the coupling with the environment that really determines whether or not we can stabilize both populations. So again, this can provide us input into our experimental studies because it can tell us we don't it's not really that we have to screen necessarily based on the properties of the individual molecules. We just have to then appropriately design the environment. And finally, I want to talk about a particular candidate for a reversible linkage. So this is a collaboration with Nick Hudd and Irina Mamedov, his postdoc, and as well as a number of undergraduate students. And the idea is that peptides, very difficult to form under plausible prebiotic conditions without enzymes. And many people have tried. Many people have tried using these cycling conditions. The yields are incredibly, incredibly low. Only dimers are formed, and it's something like 0.1% yield. So an alternative that has been has been discussed in the literature, and that we're considering, is polyesters as a prebiotic alternative to the amide bond in peptides. And this is supported by the observation that the ribosome can catalyze polyester bonds as well as the peptide bond, and that it's through an amidization reaction, one can go from ester to amide bond. But also, these ester bonds can form much more readily in water without enzymes. And so we're looking at particularly malic acid, the analog to peptides, aspartic acid, and looking at under these cycling conditions. And we first did some experiments of malic acid polymerization under constant conditions to both degrade it as well as to polymerize it. And we used that to build this kinetics model. So we have two polymers of length n and length m. Through a step condensation reaction, they can then form the longer polymer plus a water molecule. And then we can form this ODE mass action kinetics model to describe the concentration of polymers of length I for all possible lengths. We also track the water concentration, including mass transfer of water in and out of the system under heating and drying. And then we get these rate constants. So we get two prefactors and two activation energies for forward and backward reaction, as well as a mass transfer coefficient for the system. And these then we track the monomer concentration as well as the dimer concentration and the concentration of longer species. So what you see here are a number of curves. They all have the same dry temperature during the dry phase of the cycle, 85 degrees C, but we vary the wet temperature. So when it's hotter, when it's wet, we're going to get more hydrolysis during the wet phase, more breakdown. And that is, in fact, what we see here. The red corresponds to hot and the blue to cold. So what we see is we start off, we have all monomer in the system, but then that decreases as we polymerize. But then we get more of the reverse reaction during the hot, wet condition than the cold, wet condition. And that continues as expected. And then we see the inverse behavior here for the trimer plus concentration. Again, as expected, we're making polymer during the dry phase and losing some of it during the wet phase. And then we see this kind of crossover behavior, the dimer concentration. So depending on the cold, the temperature when it's wet, we either have a primary exchange here between monomer and dimer, or we have a primary exchange here between dimer and trimer plus. So it could increase or decrease, say, during the wet phase, depending on which one we're dealing with. So then we did the experiments. This is a machine we call the day-night machine that was built by Nick Hudd's former student, Aaron Engelhardt. And its core is a PCR machine to do the temperature cycling, but then we have air blowing on it at different times, and we add water at different times, pipetted in. And it's all controlled with computer. So we do GPC at the end of each day phase and the end of each night phase. So this is just an example chromatogram that we then quantitate to get concentrations. And again, just like the model predictions, we see we lose monomer during the hot phase, the dry phase. During the wet phase, we have some reverse of that, and we see the inverse behavior again for the trimer concentration. And again, we see that crossover behavior for the dimer concentration. So I think this is really validating the approach that these malic acid can be formed under these cycling conditions. So even though it's subject to hydrolysis, that we can get an initial ratcheting of the system up to higher and higher levels, and then it reaches kind of a cyclic steady state in which these polymers are maintained over time from cycle to cycle. So if these were informational polymers, all the information would not be lost every night. So with that, I would like to conclude. We have been using mathematical models in order to help us understand what regions of the parameter space of kinetics, environment, diffusion are really promising candidates for the experimental studies within the center. And then the simulations are being used to guide experimental studies. I'd also just like to mention a musical collaboration that I've been a part of through the Center for Chemical Evolution. I worked with music professor at Emory, Steve Everett, who wrote a string quartet multimedia piece about this work, and it premiered about 1.5 years ago at Emory. We've had a second concert since then. So there is a score that Steve wrote for the string quartet, but he also used computer to alter further the music. And there are also periods of computer-generated music that came from the movies that you saw. There are also narrations that were interspersed by me that I gave, as well as David Litt at Emory. And we're going to be talking about this, Steve and I, on September 19th, 7 p.m. in a science cafe at the Atlanta Botanical Gardens. So if you're interested in checking this out, timely, so it's coming up soon. And so we've had different science cafes that we've been a part of through Center for Chemical Evolution, but this is one that I'll be presenting in. So with that, I'd like to conclude. I'd like to acknowledge funding from the Center for Chemical Evolution through NSF and NASA, as well as the McDonald Foundation grant on complex systems that's supporting Andreas' work. And Sarah Walker's had an astrobiology fellowship from NASA as well. And then these are 2 of the papers from the work that I talked about. So, thank you for your attention. Thanks for coming. And I'd be happy to take any questions.
I never really heard about this sort of day-night cycling. I thought that was a really interesting idea. As I understood it, you were talking about it largely in varying the hydrolytic rate. But it also, I guess just naively to me, it seems that the hydrolytic rate and the mass transport features of the system are connected to one another.
Absolutely.
And it seems, I mean, so did that first model consider them as independent?
So in the first model, we had these 2 distinct phases. So there was only polymer formation in the first phase, and then we didn't have any evaporation in that system explicitly, but we had, in the wet phase, we had the hydrolysis and the diffusion. Now, but in the polyester experiments, in the day-night machine, everything's going on at the same time. And we did include that, so maybe I'll just go back to that slide. But so we have, this is all going on in the PCR machine. So what we did is we added water, and then we heated up the system. Or we heated it up, we added the water kind of at the same time. So the day phase, the dry phase, went on for 18 hours. And the water had evaporated within about the first 15 minutes. So in the first phase, we add the water, probably nothing happens until all the water evaporates. Then we get polymerization during the next 17 plus hours. Right? So we can kind of decouple those phases. However, you can see here these kind of little blips at the beginning of the wet phase. That's because we add the water, and then we get some polymerization as well as hydrolysis kind of going on at the same time. No, actually, sorry. We get the hydrolysis going on here, but we do get, once we've made some polymer, we add the water during the hot phase, and we get some depolymerization there. So that's a coupling between the phases, because it does take time to evaporate that water. And so we have this mass transfer coefficient to describe that effect. So you're right, all of these are, the hot and the wet and the dry and the cold are all going on. And in fact, they're not going to be, these experiments also is very discrete. We add the water at the same time as we change the temperature, but you're really going to have something more like sinusoidal profiles in a geochemical setting.
Yeah, and I guess I was kind of interested in the idea of sort of like bulk wetness versus local wetness, particularly if it's a condensation reaction. It seems like you're locally producing the hydrolytic agent
for sure
in the polymer setting. So
right. That's a really good point. And I mean, I think that's why I like the spatial modeling that a lot of, so we did some of the well-mixed model, we did some of the spatial model, but these kinds of effects are something that you need to explore spatial heterogeneity. And so you could then have local, you'd have water diffusing around on the surface as well as evaporating too in those spatial simulations. So, I mean, we had the assumptions that are made in the spatial simulations are, like I was talking about with Valeria, are very simplifying in many ways, because we wanted to be able to explore the key effects with a number of parameters that, I mean, 5 is already a lot to explore. We explored, one two-dimensional slice and another two-dimensional slice. We still didn't do the whole space. Now, we could have done the Latin hypercube sampling to explore the whole space. That would have been maybe a more efficient way to go to explore a larger space. That's what we did in Andreas' study, because we had 8 parameters in that case, and that's just too hard to explore with two-dimensional slices. But anyway, these are all effects that we could add into our spatial simulations. The real question is how to parameterize it and how to not have too many parameters. And if you have an experimental system, then you can model that system, which is the case with the polyesters. But if you're trying to generally explore the parameter space to identify what experiments you should do, then it's harder to do. But I actually think this Latin hypercube sampling of a higher dimensional space is a good way to go, and not something that I've really seen too much before. It's a lot to do. Any other questions?